Scientists use mitochondria to reveal cancer cells to the immune system and destroy them. In a surprising discovery that could revolutionize the way we treat cancer, American scientists have found a way to use a cellular organelle called mitochondria to make cancer cells more visible to the immune system. Mitochondria are the powerhouses of cells responsible for generating energy. Researchers from the Salk Institute in San Diego have developed a new way to improve the body's ability to recognize and fight cancer cells, promising to make immunotherapy more accessible to a wider range of patients. The study was published in the journal Science and featured in Newsweek. In the study, the researchers made changes to mitochondria to restrict energy flow, which led to the accumulation of a substance called succinate. Succinate has a number of roles throughout the cell, including regulating gene expression. By increasing the concentration of succinate in the cell, the researchers were able to increase the expression of certain genes that participate in communication with the immune system. The immune system's sentinel cells patrol our bodies, and our cells display a set of molecules on their surfaces that act as a kind of molecular identity to signal that they are healthy. These molecules are sampled randomly from inside the cell and are anchored to the cell surface by special structures called glycoproteins. Rewiring mitochondria to produce higher levels of succinate leads to an increase in the number of glycoproteins on the cell surface, meaning that more components can be taken from inside the cell. This means that if there are any rogue mutations floating around inside the cell, they are more likely to be picked up by the immune system. In other words, it becomes harder for cancer cells to evade our immune systems. These changes essentially turn the tumor into something that the immune system can recognize, said Susan Cake, co-senior author of the study. This could have a very big impact in the field of immunotherapy. Tom McVicker, an expert in reprogramming mitochondria in cancer at the University of Glasgow and the Beatson Institute for Cancer Research in the United Kingdom, said, "Mitochondria play a multifaceted role in cancer. For example, mitochondria drive the biosynthesis of macromolecules, such as lipids and DNA building blocks, that are essential for the growth and spread of cancer cells. Mitochondria are also important for the survival of cancer cells in harsh microenvironments and in response to cancer treatments. They regulate the cell's internal balance, cell death pathways, and inflammatory pathways. How to target this rewiring of mitochondria to specifically target cancer cells remains a challenge. Cage said that these specific metabolites can be delivered to the cell in different ways, providing several potential drug delivery routes. We have shown that we can provide succinate to cells externally and get the same effects, so succinate can come from either outside or inside the cell," she added. While more work needs to be done to translate these findings into clinical practice that can be applied to patients, this discovery provides a new and exciting avenue for future treatment solutions.